Hi everyone, and a very good evening to everyone, and thank you for staying on. Um, we had a bit of a technical glitch in the middle, so I'm extremely sorry for the delay. So this is Aishwarya again, and I hope everyone can hear me as well as see me. In case you're not able to, please put that on the chat box or something. Okay, so today's topic is tissue engineering and regenerative medicine. This is ideally the main topic, and our yesterday session was basically on how we would start with 3D bioprinting, what is a general idea of 3D printing as a tool, and from there we will go down to this next topic. So I will share my screen now and present to you all. Yes. All right. So the topic is tissue engineering and regenerative medicine. And as always, a quick introduction. My name is Aishwarya Shroor, and I'm a technical specialist at Altum Technologies. We are a 3D innovation platform company, and we mainly work with several solutions, including the Sol systems, CATIA, MSC software, Stratasys, Arctic 3D, BioVia, as well as selling solutions. So, I think I've already given you these introductions that we've been a decade-old company and we have quite a large set of clients, partners as well who we work along with, and this is how we bring forward our solutions. So, coming down to the main topic and how tissue engineering comes into picture. Ideally, when it comes to any form of deformity, we would usually see that 3D printing or any sort of methods we would use have a certain layout. So, when we talk about the treatments, the general treatments that you would see in terms of any deformity, say any deformity in terms of say any accidents, any ailments, any congenital diseases, anything of that sort, you would go with artificial prosthesis, which is basically a definition in medicine where a prosthesis or prosthetic implant is an artificial device which is meant to replace a missing body part which may be lost through trauma, disease, or any conditions present at birth. Now, these processes are intended to restore the normal functionality of the body, that is again missing part of the body. And then you also have conventional treatments apart from prosthesis, which was initially developed in terms of technology advancements or any forms of treatments that people have. The other being surgical reconstruction. So, when you come to surgical reconstruction, this is basically repairing parts of your body affected by defects. So, here you have surgeons working along, trying to do a surgical procedure on you, trying to repair a specific defect. For example, a cleft lip or any palate repair, or any breast reconstructions after any sort of surgery or tumors. And this is a basic example of it. So, while you have these conventional treatment methods, we are trying to find a specific form of treatment that can be less painful, less amount of comorbidity would exist. So, the problems when it comes to these conventional methods, it would be artificial prosthesis. When it comes to artificial prosthesis, you are subjected to mechanical failure upon long-term plantations. You have rare integrations between the host and the tissue site, and then you have triggers due to any immune responses. So this comes under the general processes, and then when you come to the surgical reconstruction, you have failure due to any replacement of any functional part, development of any malignant tumors, surgical complications, morbidity at donor side. So these are very effective methods that we would use when it comes to treatment of any diseases, especially accidental or trauma cases. But there are also some backlogs to these or some negative impacts of these, although they work the best. So, keeping that in mind and having to create something more of a solution to this problem, that is why we are dependent more on artificial means, and rather let's go on something more on a biological or a natural way, is how you have the concept of tissue engineering. So, tissue engineering is ideally an interdisciplinary field, and this applies the principles of, say, engineering, life sciences, and the idea is to develop biological substitutes in order to restore, maintain, or improve tissue functions. So, tissue engineering is ideally applied in the area of regenerative medicine. Basically, what you're trying to do is you are trying to regenerate, regrow a part of your body which is not functional with something that can induce function into it and allow the growth of that part in your body. So, this intends to stimulate the replacement of dysfunctional or lacking tissues. So, you are trying to stimulate or induce a form of growth within that part of the body. Say it is bone, maybe cartilage, maybe skin, say in terms of wound healing, or any other examples I would go about. So, tissue engineering ideally has few basic components or few basic functionalities within it, and that would start with scaffolds, and then it comes with cell type, and there comes chemical and physical factors. So, if you're talking about scaffolds, scaffolds are known to provide porosity. They're cyto and tissue compatible. They're quite bioactive. They allow cell attachment and migration, and mechanical properties as well. Cell type is basically the routine where you're trying to select the type of cell. You are making more relevant cell models, checking for better interaction, better signal pathways, integration of flow. And the third being chemical and physical factors. So, the chemical and physical factors would be the environment in which your culture or your tissue is growing in. That is with the help of medias, a specific humidity, a specific temperature, a contamination free zone, or an aseptic zone. These are some things that you would essentially require when it comes to creating any form of tissue engineered model. So, when coming down to the types of cells, we have embryonic stem cells, which are basically pluripotent stem cells, which have an infinite self renewal capacity and have a very high potency. So, when you look at embryonic stem cells, they are taken, at a site which is the blastocyte state, which is the most earliest state. They are cultured, and then they are allowed to differentiate further, giving you more parts. That is, say, giving the three lineages: the ectoderm, the mesoderm, and the endoderm. So when you go to the ectoderm, you have maybe the skin or the nerve. You go to the mesoderm, you have bones, muscles. You go to the endoderm, you have maybe liver, thyroid, and so on and so forth. Then comes the induced pluripotent stem cell. So these are basically, the word induced is used here. So you are reprogramming a type of cell which is already in its state. So it is in a differentiated state or maybe in a midline state where you let it go through some set of programming. Programming would be in terms of chemicals, in terms of physiological factors, or any other factors that could contribute towards reprogramming your cell so that it goes from one form to maybe the initial form or the later form. Then there is adult stem cells. Adult stem cells are mesenchymal cells, hematopoietic cells, neural cells, etc. And they have the tendency of being quite directly used in a lot of cases. So there is a lot of work done where mesenchymal cells have extensively been used in terms of creating neural or somatic cells or stromal cells, etc. And then you have differentiated cells. So differentiated cells would be just directly using maybe cartilage cells or directly using bone cells, taking it from its site, isolating it from its main site, and then using it. So, when you come down to taking the cell type, there are factors which contribute towards this. For example, the targeted number of cells. Having a right amount of cell will also give you the right amount of biochemical production, which will in turn induce the amount of regeneration that needs to happen at the site. Then there is a number of passages. So passages are basically when you have cultured your cell, a stem cell, you need to find the lineage, the age of it. So say example, at the time of its source, it has say passage 1, and then you keep on growing that, you keep on differentiating it, or you keep on culturing and growing it over time, is when the passage numbers go on increasing. So it is always ideal not to have the higher amount of passage, keep it on the lower side, because age is something you need to consider when it comes to cells. Then the time of culturing is another point. Then you would consider whether what is the state of the cell and what is the storage conditions of the cell that you have taken. Then comes scaffold. So scaffold would be the main building block or the main foundation of what you would create. So you would have biocompatible cell attachment and migration retention of cells and biochemical factors, which is what the scaffold creates. It will allow diffusion of the cells. You would have vital cell products being created. Then you have pore interconnectivity. You'll have behavioral modifications that can happen in terms of how your scaffold is, giving you the better amount of mechanical strength and also the environmental conditioning required for a job. Now, when it comes to creating scaffolds, you always require some sort of material, say artificial or natural material, which can create this scaffold. Now, this scaffold is something which will have the capacity of enholding or enclosing your cells within it, and they would use biomaterials. So, materials, biomaterials, biological materials would be used here, and they are used to develop scaffolds. Now, these tend to provide a template for the cells to organize and restore structures and functions to damage dysfunctionality. You have natural materials, say for better biocompatibility, low toxicity, cell mediated proteolysis. Um, Examples of natural materials, they would be collagen, fibrin, gelatin, chitosin, alginate. Synthetic materials, they would be, say, having a better amount of degradation rate, controlled mechanical property. You can easily process these materials and they're easily modifiable when it comes to using materials like PLA, PLGA, PCM. So, standardly, when you come in the bioprinting space, the kind of materials you will often hear would be collagen, fibrin, gelatin, and alginates. So they are easy to work with materials, and also you will have PCL, PLGA, most often used because they are easily blendable, and they're also easily blended with bioceramic materials. Say example tricalcium phosphate or hydroxyapatite or any material which can effectively work in creating a bone like structure. So, the biomaterials that you have, or what you require when it comes to tissue engineering in general, or when it comes to using it in the space of bioprinting, would require strength, elasticity, biostability, lack of toxicity, in some cases, magnetic property, a better host response, mechanical stimulation, correlation between biocompatibility and printability. And there's also a spatio-temporal arrangement that is required. You cannot use a material which is of a very high polymerization index, because you don't want the spaces to be completely compact. There's a need to have good amount of porosity, good amount of spacing, so that the cells also can bind in every direction, that is in a three-dimensional manner, and it can also allow growth of these cells. So, these biomaterials or tissue engineered materials that you have can either be injectable, they are supposed to have appropriate mechanical and biophysical properties, like I mentioned. They should ideally release the biomolecules that you require. They should also have a standardized degradation kinetics. And when you're degrading a material, when a material is degrading, it should also be important that there's a lack of toxicity over here. The architecture, morphology, and whatever material that needs to deliver it aside, that is the biomolecule, should also completely pass through the pores of your material, should also be easily integrated. So, biomaterial, the specific architecture, the biomolecules, this will actually stimulate the niche cells. So, having the right amount of material which can give you the good biocompatibility, which will aid the cells to grow better, which will have a good structure, that is a good physiological environment, as well as the secretion of biomolecules either by the material itself or the material or the stimuli of kept it in, will allow better regeneration. Next comes the chemical and physical stimuli. So, in the chemical and physical stimuli, like I mentioned, it could be the culture media, it could be the temperature, it could be the incubation, the bioreactor, time of differentiation and culture, the technology, the amount of cell signaling, and the cell cell interaction. And this is very crucial when it comes to any aspect of working with cells, because we have to ensure that if there is good interconnectivity, there is a better amount of production, there's better amount of stimulation, better amount of conduction as well as induction in terms of the functions you're trying to achieve. Now, when you come down to the scaffolds that are created and how tissue engineering would help create a specific type of model for where you want to use it, you would have several methods. So we have leaching method where you have solvent casting, ice particle leaching, gas forming. Then you have microsphere methods where you have bead formations, particle aggregations, phase separation where you have freeze drying of material using thermally induced phase separation. Then you have some widely used methods like fiber spinning, that is electrospinning, nanofiber electrospinning, microfiber wet spinning. Then you have decellularized matrix. This is basically taking a tissue and making it devoid of cells, removing the cells of it so that you have the ECM. End of the day, the process of creating a scaffold is creating something that can act like an ECM, that is allowing a tissue space and then having cells being added to it, which can give you that kind of support when it comes to regeneration of the cells. So these are conventional methods, and these methods are definitely time consuming when it comes to creating the final scaffold. Not to mention, there is also other processes involved initially when it comes to even creating the initial chemicals or finding the right standardized chemicals, creating the scaffold, standardizing the scaffold. So every process has its own pros and cons, and while these are scaffold creation methods, there are some disadvantages that exist. That is, they take a long duration, they're quite costly because you have to use multiple types of equipments to carry out the process. There is uneven distribution of cells. The reproducibility is a main issue when it comes to trying to create scaffolds. Say example, I have standardized a method. There'll always be batch to batch variation, there will always be differences in the kind of pressure exerted, the solvent concentration, the molar concentration, and this might cause a lot of delay, and you might not get effective scaffolds that you require. And the major disadvantage of conventional methods is that it fails to replicate geometry. So we are trying to create an environment which is more physiologically relevant, a more tissue specific environment, or a more tissue specific geometry. And in the end of the day, if you're just trying to create a basic circle or a basic triangle or any structure, as a matter of fact, it might not always give you that kind of effect, because you also need to play around with the structure of it, also the porosity of the material. So, conventional tissue engineering would require, say, example, creating a scaffold or creating any type of structure. Here, you are going to seed it with cells. Now, you have created a structure. To that structure which is created, you seed in cells, and basically, seeding means taking so many amount or so many concentration of cell suspension and just dropping it over the scaffold you have created. Now, this will definitely cause wastage of cells because they don't entirely go and bind intact within the crevices of the scaffold you have created. They have not completely stayed in the structures that they need to be in. And also, you will face some amount of gaps in terms of seeding the cells, putting it into an incubator, doing the rest of the job. So, keeping these disadvantages in mind, you have 3D bioprinting. Again, I won't dwell too deep into what 3D bioprinting is, because I think um, in our previous session we've already had uh, you know uh, my introduction on 3D bioprinting. So again, a form of additive manufacturing which ideally uses bioink materials to print living structures in a layer by layer fashion. And the idea is to mimic the behavior of natural living systems. So it's an integration of 3D printing, tissue engineering. So you have cells, bioinks, culture media. I've also mentioned of the extrusion printing as well as the digital light processing. So this is a quick glimpse of the processes. And this is the DLP. So further, I will explain how each of these techniques will provide that much amount of value in terms of creating specific designs. Like I also mentioned before, I'll still repeat this bioprinting steps because this is how each application would benefit from the steps. So, anatomical data is one way how you can get the idealistic structure of what you want to create. You have MRI scans, you have CD scanners, which will give you the design. 3D designing is basically, say, example, for some reasons or some cases, you would take segments of the standard MRI images to get a customized model, or sometimes you just want to create a basic design. So you'll have the 3D design, and then you have crucial points being material selection, cell selection, extrusion, DLP printers, again, the methods of or the types of printers you would use, curing, which is again a cross linking method, incubation, and in vitro tests and applications. So, when you have tissue engineering involved, you will always have the first step being in vitro test, the next being in vivo test. So you will definitely go through the process of in vitro testing your model. That is testing it in the lab space, doing live data analysis, compatibility tests, checking there is a biochemical marker. You want to do immunofluorescence staining. You might want to do DAPI tests, also sometimes confocal tests, and then comes down to the application. Say we have to go through the stages, which is trials, in vitro testing, trial 0, that is preclinical testing, then you have phase 1, 2, 3, right? In this manner you go on testing. So we are at a state where we have in vitro testing, we have animal testing. So these two tests become very crucial in terms of understanding the capabilities of tissue engineering and how it could be further used when it comes to a larger set of people or a larger set of group. So bioprinting is a potential case where you could have organs created in near future, or you could have easy grafts, easy skin material, and also there are companies like L'Oreal which produce skin for in vitro testing. You have some companies that create wound healing structures. Some companies that create materials together that can easily allow making any form of model when it comes to wound healing or bone regeneration or any other example of that sort. So this is the first example, being a simple example and most widely used example. This is, at least there is recognition quite a lot in India where researchers have successfully been able to create cornea models. So here the basic idea is getting the dimensions of the cornea structure. So you have to take the digital model of the person of the cornea, and you have to take the corresponding model of it, that's a 3D structure, create the code, something called a G code with the infill, then selecting the material, saying biomaterials, using the cells, blending them at a right proportion, and then you would go to the process of printing it. Now, when it comes to printing this, you would have extrusion based printing as well as DLP based printing. So, when you have extrusion based printing, there is a possibility that you might require a support. Support being a material, say, for example, if I'm trying to create any sort of curvature at the site with a material that could be slightly soft, I would require this cap as a support structure to print anything over it, to hold a structure which is concave or convex, right? That's where support is required. And there are some materials which will also not require any sort of support. um, and when you go with a method like DLP printing, right, DLP bioprinting, you would not require a support structure because this is supposed to be a print of high resolution, and also the materials tend to be quite rigid when it comes to this. So you have prints of cornea done on the DLP method as well as on the extrusion based method. The final stage will obviously be checking the physiological factors, testing the lens which has been created, the cornea which has been created, and this requires a aqueous medium. So naturally, we know that we have the aqueous humor, we have the eyes itself with several sections, and there is fluid retention or fluids that flow through your eyes. So naturally, it becomes important for you to culture whatever corneal structure in a liquid medium. So this is again example, which is again a live example where companies are creating corneal models, doing the preclinical testing, doing the in vitro testing, and have also succeeded to a point. So we are steps away from trying to put this out as a realistic, a real treatment coming out in the market quite easily. Then you have cartilage constructs. So this is example of again in vitro, an in vitro to an in vivo model where they have tried to take tests of autologous cartilage, 3D printed cartilage, 3D printed, basically 3D bioprinted cartilage, and also scaffolds with cell seeded in it. So in this, you will see that there is good amount of regeneration in comparison to the 3D printed cartilage as comparative to maybe the native, that is the autologous, or the PCL, that is a cell seeded cartilage. So you'll see high amount of regeneration, and also sometimes what happens is when you use native, that is autologous cartilage, there is a lot of scar formation. There can also be a lot of thrombosis at the site. Using cell seeded models can also cause a bit of a dysfunction. So if you try to compare between all three of them, we find 3D bioprinted models more effective in terms of interconnectivity, in terms of the networks, the interactions, and also in terms of inducing good amount of cell regeneration. This is an example, and this is an example again in India. This is by IIT Guwahati. So here they have used silk as a material in combination with gelatin, and they have created this bio ink material, which is known to be efficient in terms of creating meniscus models. So they have tried to mimic a tri-layered meniscus for potential patient-specific implantation, and the work is going towards that. The idea behind creating a meniscus is for the development of knee osteoarthritis. Have a patient with osteoarthritis would have his cartilage worn off over time, and having a replacement becomes very essential. So instead of replacing the whole knee joint, putting an artificial material, we would rather use a bio-artificial, that is 3D bioprinted cartilage model, which can be effective. So here they have built the architecture quite well, and how they have built the architecture is also by taking patient-specific data. That is using MRI, CT scans, taking segmentations, taking the specific segment of that, trying to create the 3D design model, and then printing that model with factors of good viscosity of material. And also, when you're creating the scaffold, you have to check for optimum swelling, degradation, mechanical properties, and also how the cells have been distributed. So here you have a good amount of compatibility both in the in vitro as well as the in vivo realm. So there is gene expression profiling that they do, biochemical quantification, histological studies that have to be carried out, and eventually there comes a point where they have approved of using the specific material for meniscus development. And from there you are also a long way into um, directly getting a patient handheld meniscus as such. The third example is basically a heart patch. So here they have used gelma as a material, and this is a comparison between using cell therapy versus using just the material versus using any ECM material, and also using a bioprinted material. So the bio ink here is Gelma, and here they have seeded it with cardiac cells. And the idea here is to repair ventricles, maybe in terms of congenital diseases, maybe in terms of repair after myocardial infarction. So they've created patches to put it at a site where they will be able to do the testing in such a manner that there is good amount of regeneration at the site of infarction, or at the site where the ventricle has not developed. So, in terms of heart in tissue engineering, we have a guest speaker coming tomorrow, Dr. Shreya Malhotra, who will give you more insights into what cardiac printing is, what cardiac tissue engineering is, and whatever she has experienced herself when it comes to working with heart cells. So, I hope I see you there as well. And from here, I'll just go down to basics, our general solutions, how does the machine work, what are our other solutions. So we have the incredible system, and then you have various other solutions. So incredible being the dual print head system, you have Incredible Plus, we have BioX 6, which is a more complex machine, more aseptic machine. We have industrial grade machines like the BioMDX, the BioMDX Plus, we have Luminex Plus, and the Quantum X Bio. So the Luminex Plus as well as the Quantum X Bio are highly recommended for microfabrication, say microneedles or miniature structures, creating implant devices, drug delivery devices, graft models. All of these become very easy in terms of resolution, size of printing, any biological sample as well. So the uniqueness of Luminix Plus is that you can print with cells when you're using the DLP. Most of you might not find it surprising because generally DLP method, which is a engineering method, is used to just print materials. But this is a very unique machine known to print with cells because it has the ideal amount of space, temperature, the light which falls on it, and easily allows vasculature when it comes to printing with cells. So, a quick glimpse on what the BioX system is, or the what the extrusion based system is. Okay, so this was the BioX system. Now, a quick view on the Luminex Plus system. So here, this is a touch interface system. So you have something called a build platform, a VAT, which keeps the material that you want. So you will easily place the material that requires to be printed. And within a few minutes, you will have the structure that you want printed. So yeah, another solution that we have is the BioVia. Again, it's a biological modeling and simulation software. In case you want details of this, we have more of this on our Atom Technologies website. We also are available on YouTube channel for more descriptions on the product. And so this is mainly capable of protein engineering, antibody modeling, simulations, ADME studies, X-ray analysis, and it is a solution which is in the bioinformatics and the chemoinformatics realm. And yes, so just a heads up, tomorrow 7 p.m. we have Dr. Shreya Mehrotra who's going to talk about 3D bioprinting in cardiac tissue engineering. And she is a postdoc fellow from IIT Kanpur. And I hope I find more people joining us there, asking more questions, and also supporting us with, understanding 3D bioprinting and spreading the word. So thank you so much. As usual, please find us on Facebook, YouTube, LinkedIn, and keep following us. And if you have any queries, you can easily find us on altumtechnologies.com. So just a second. Yeah, so I will take questions, if any. Okay, so this is a question which I might not have an answer to. It says, does ANSYS support have any module which can simulate 3D bioprinting? So I think we will take this personally, also discuss more about it. Do we have any more questions? Yeah, so I don't see any questions for now. Thank you so much for staying on. I hope I see more of you, more of people tomorrow, and Have a great day. Yeah. Thank you so much.